Good afternoon, everyone. You are all most welcome to this fourth webinar series in gene therapy under the EBCTP Capacity 2023 project. This project has 5 partners. There is a Joint Clinical Research Center where I'm based, the National Drug Authority, the National Council for Science and Technology, Muhimbili University in Tanzania, and then University of Coimbra in Portugal. All of you are most welcome. My name is Francis. I'll be your moderator today. And my background, I'm a medical doctor specializing in internal diseases and mostly especially diseases of the blood. So we are based here at the Joint Clinical Research Center. We have had very excellent speakers in our previous sessions that we've had that have introduced us to gene therapy. And today we have our main speaker of today is Dr. Kevin Dokzen. Dr. Kevin is a biophysicist, and he initially trained in Johns Hopkins and then did his PhD in Berkeley, California. So this afternoon, we have a very exciting topic. He's going to walk us through the next generation of gene editing. We are going to hear more issues on base editing. We are going to hear about prime editing. What difference does it make? We are going to hear about when gene therapy is being done. Do we focus on the gene itself? Do we focus on the proteins in which the gene is wrapped? And we focus on other parts of DNA that actually are not important for the gene to influence or silence or activate the gene. What are the methods available? Do we have a one size that fits it all? Because some of the genetic diseases have, very small, simple genes. Others have very complex genes that have to be addressed. We are going to hear about the methods that are available, the ones that are using solutions from nature, using enzymes, from bacterial enzymes, and then I think in the past we also had editing using viruses. I'm sure we are going to hear some more in detail about gene therapy or gene editing using nanoparticles. We are excited, I think, looking forward to hearing about the various types of nanoparticles, whether they're lipid or other forms. And I'm sure we're also going to hear about modifying large chunks of a genome or DNA versus modifying just small bits, whether one needs to break up the DNA to make a change, it needs to undertake the gene therapy or not. So we are going to hear how the story of gene therapy has moved and where it is moving. And perhaps we'll also hear more what influences the method that is going to be used. And I'm sure we'll get some insights from Dr. Kevin where he thinks that this form of therapy is going forward. I'm sure we are going to hear about some of the successes and maybe some of the existing challenges with the methods that we're going to hear. But towards the end, we also have an evaluation. Please, I would encourage all of you to fill in that evaluation form. It will help us as we move forward. So without much ado, Dr. Kevin, you're most welcome.
Great. Thank you so much for the generous introduction. Like was said, I'm Kevin. Did my PhD at Berkeley with Jennifer Doudna, and then after that, I worked for a little bit at the Innovative Genomics Institute at Berkeley, and then did my postdoc at the World Economic Forum, which is where I got plugged into the Global Gene Therapy Initiative, and I work with several of its members to create a white paper, which I'll show on the very last slide. And then currently, I'm contracted to an agency in the US that works on healthcare innovation. Um, and so I've been following a bit kind of what's on the cutting edge of gene therapy. Where are people taking exciting innovations from academic labs and turning them into companies? And then how are companies trying to make sustainable business models out of these gene therapies? So over the next few slides, we're going to cover a lot of ground. Um, I'm kind of going over trying to do a very broad landscape review. But if you want kind of a deeper dive into a particular topic, I have my contact on the last slide, and reach out to me and I can provide some resources. I'll also be kind of highlighting different companies along the way, mainly because they offer great graphics, and I think they give a good example of where the field is right now. But I'm not like endorsing any of these companies, just using them as examples. And so we'll begin with kind of laying the foundation of CRISPR-Cas9. And I know you've already heard a bit about this. I'll just give one brief slide. Uh, so it's important to note that there's two main mechanisms to repairing a double strand break, right? So you engineer your Cas9 using the guide RNA to target a genomic sequence of interest. And then after it cuts both strands of the DNA, you have non-homologous end joining, which can insert a couple of nucleotides or remove a couple of nucleotides, which will essentially kind of break that gene. And this could be useful if you have a gene that's mutated and it's creating a toxic protein. And NHEJ, which acronym that's used for it, is active throughout the cell cycle, and works well in non-dividing cells. Homology-directed repair um, it uses a homologous DNA sequence. Sometimes that can be the sister chromatid of the cell. Sometimes that DNA sequence is actually provided by the researcher, uh, and it is inserted at that site of the double strand break. And this can be used if you need to introduce a healthy copy of the gene. But HDR is only kind of efficient during certain parts of the cell cycle when a homologous template is available, and so it doesn't work really well in non-dividing cells. And so there's limitations to kind of this powerhouse tool that people have been using, this CRISPR-Cas9 enzyme. It can have off-target editing and cuts. It's dependent on a PAM sequence, which I won't go into, but that just means that um, it, it's difficult to target, every region of the genome. Um, NHEJ and HDR can be just inefficient. There's inefficiencies in the system. Delivery challenges. Some of these Cas9 proteins are quite large. And then immunogenicity. So these proteins derive from bacteria, and so they can induce an immune response in the human cell when added. So a lot of the researchers saw these limitations and they wanted to circumvent them, to deal with them by engineering Cas9 in some pretty unique ways. And so one of those ways is by base editing. So base editing, it's important to note that it uses a version of Cas9 called dead Cas9. And so to make a dead Cas9, researchers mutated two of the amino acids, an aspartate and a histidine. And so this Cas9 will use its guide RNA to find a genomic sequence of interest, and it'll just sit there. It won't make a cut. And then they've made two main versions of this base editor. And so attached to the Cas9, they add a deaminase. And so in the case of the ABE, or the adenine base editor, they take adenine and then they eventually convert it to a guanine. And then in the case of the cytosine base editor, the CBE, they take uh, a cytidine and convert it to a T. And so that's really exciting. You know, in so many instances, you really just need to change one letter in the genome to correct a disease. And so base editing, um, some really kind of ways it circumvents the limitations of the Cas9 that makes double-strand breaks is that um, it can be a bit more efficient. So, because it only needs to make a single base change and doesn't have to go through the process of uh, NHEJ or HDR, these DNA repair mechanisms, it ha can have a pretty high efficiency level. Um, it doesn't require a double break, like I said, so that can just prevent kind of rearrangements in the genome and other um, uh, outcomes that you don't want for safety reasons. And it works in a variety of cells, including non-dividing cells. So unlike HDR, you're not as limited. But there are cons. So like I just mentioned, you have the C to T and the A to G. So there's other substitutions like a C to G or an A to D, A to T, that as far as I know, we can't do. Maybe that's out of date, but just in general, there's a limited ability. There are bystander edits. It's not 100% targeted on a single base. Sometimes bases next to it can also get mutated. And there's delivery challenges, right? So we added a whole nother enzyme, the deaminases, to this Cas9. So now you have to deliver all of that into a cell to make a base edit. So you're going to need a bigger delivery vehicle. And then there's still immunogenicity, right? So these Cas proteins and some deaminases are still foreign proteins, and so you can still have that negative health reaction to it. But the main question is, has this entered the clinic yet? And the answer is yes. So as of early 2025, a total of 15 trials have been enrolled in clinicaltrials.gov involving 11 base editing drugs that specifically target, I'm going to mess up these names. I'll just say GCD, and then leukemia, sickle cell disease, beta thalassemia, Duchenne muscular dystrophy, and hypercholesterolemia. And I'll just highlight one or two cases. So this one's from Beam Therapeutics. This Beacon study is evaluating the safety and efficacy of what they call Beam 101, which is for sickle cell disease. And so for Beam 101, the target that the base editor is going for is the promoter region of the HBG 1 slash 2 gene, uh, which normally produces fetal hemoglobin during human development, but is switched off in adults. And so the researcher will engineer the guide RNA to target the promoter of that gene, and then the edit, that one letter change, is designed to block the transcriptional repressor protein BCL11A from binding to that gene promoter. And so in total, what they're trying to do is by preventing the enhancer from doing its job, the therapy effectively enhances or switches back on the production. So you're enhancing, you're down regulating this repressor, That all sounds a little confusing, but essentially the main outcome is that you want to turn back on the fetal hemoglobin, which is normally turned off in adults. And what's kind of exciting is this was done. Here's just an example from Boston Children's Hospital. Brandon started this process in October 2023. He was infused with edited cells in December 2023, and following the procedure, had fewer crises or no crises for a while. I don't know the current status as of now, but it was just a great example of this approach working. And then I'll give another example real quick from Verve. So Verve, they were interested in cholesterol. And so specifically, they were interested in low density lipoprotein cholesterol, or LDL-C. And I'm listing two right here, Verve 101 and Verve 102. They're essentially the same thing. The only difference is the delivery nanoparticle that was used. But they each used the same ABE, uh, and they're both targeting the PCSK9 gene. So what's the PCSK9? It's a protein that binds to and degrades this lipoprotein receptors on liver cells. So this prevents the receptors from removing cholesterol from the blood, leading to increased cholesterol levels. So what was really exciting is that they saw this dose-dependent response. The more of this base editor they added, they saw a decreasing amount of cholesterol in the body. So they did see this base editing work. Targeting the PCSK9 gene did lead to lower cholesterol. And so putting these two case studies side by side is interesting because um, I think one is an example of maybe a condition that impacts a fewer number of people, and then the other, cholesterol, is maybe a little more people. So you have companies looking at a wide spectrum of conditions that affect a wide diversity of people. And hopefully we see that more and more, these technologies being used to address a wider and wider range of conditions from both rare, ultra rare to common. And I think it shows the potential of base editing as a really useful tool. And it's really surprising, I think, the number of conditions that can be treated using just a single letter change. So now we're going to pivot to prime editing, which I think is a super exciting invention. It's a little more confusing than base editing because you're not just making a single letter change. They've engineered this really long and kind of wonky, what they call PEG RNA. So these prime editors are engineered to consist of a reverse transcriptase fused to a Cas9 nickase with a PEG RNA. And I have a little video to kind of visually show what's going on because I think it's a little easier than a static image. But from this cartoon, you can see that the Cas9 is bound to this reverse transcriptase, and reverse transcriptase turns RNA into DNA. And then the pegRNA, the reason it's so long is because this some that stretch of the pegRNA where they have that little blue dot that says replace, that's actually going to be used for that reverse transcription. So the reverse transcriptase will read that, turn it into DNA, and is what will be used to be inserted into the cell. And this word nickase, we haven't encountered that yet in this talk. That's similar to a dead Cas9 in that the researchers mutated a Cas9, but instead of making two mutations to prevent the double-strand break, they only made one. So it just nicks, it just cuts one of the DNA strands, not both. So yeah, the PEG RNA is a modified guide RNA. And like I said, it's longer and it's designed to have that uh, RNA that will get transcribed, reverse transcribed into DNA that you want to insert. So I'm going to play this video. Let's see. So this is from Prime Medicine, the company. So here's your Prime editor, this Cas enzyme, and then it's bound to that reverse transcriptase. You can see it's quite a large macromolecule when they're both bound. And then there's the Cas9 protein bound to the PEG RNA. And you can see there's a long stretch of it sticking out of the Cas9 sequence. And that's what's going to be used to reverse transcribe and turn into the DNA that'll be inserted or added into the genome. So now this Cas enzyme is using its guide RNA to search all around the genome for a matching DNA sequence. You can see the little histones in there. And you can see multiple of these Cas RNAs, right? You don't just add a single protein into the cell, you add a bunch of them and they're searching everywhere. And that red blinking base is what we're hoping to change. And so the guide RNA, the Cas protein unwinds the double strand DNA. The guide RNA finds that complementary sequence. And once the complementary sequence is identified, which is shown in that blinking uh, strand, we'll see the nick happen, right? So it's right there. One strand is cut. So I always find that amazing, the precision by which this happens. And then here we go. The PEG RNA is going to loop all the way around, and then the reverse transcriptase is then going to read that RNA and then turn that into DNA. And when they were engineering this whole system, right, they had to get that linker length between the Cas9 and the reverse transcriptase just right. Can't be too short, can't be too long. So then once it's turned from RNA into DNA, it goes whoop back into the double helix, and then endogenous proteins that just normally exist within your cells, come and cut off that extra strand. And then boom, easy. You have your corrected gene. And so I hope that was a bit useful. So prime editing in general is here we go. There we go. That's the correction. So the why prime editing was so exciting when it was first announced is because it can you still don't need a double strand break, right? You're just making a nick. So you're avoiding just cutting your DNA in half, and which can lead to potential rearrangement of the chromosomes and other really harmful um, uh, things happening to the genome. Uh, but like base editing, you can avoid cutting the genome entirely. You just need to make, in this instance, just a nick. But unlike base editing, you can make a much larger spectrum of changes to the genome, and you're not just limited to a single base. I forget how long exactly the PEG RNA can be, but it can be beyond, 10 bases. It can't be super, super long, and we'll talk a little later about technologies that are enabling really long insertions of DNA. But at least this one gives you more options than base editing. But there are still cons. It's still not entirely 100% efficient. You're still delivering a very large macromolecule, this Cas9 tethered to a reverse transcriptase, and the PEG RNA is very long. PEG RNAs are a bit more complicated to design. That being said, there are more and more better tools to help design for researchers. And then and they're still off targets. So is this in the clinic? And the answer is yes. So that's also really exciting. Here's this chronic granulomatous disease. So Prime Medicine has targeted this rare disease, although it's estimated to occur between 1 in 100,000 and 1 in 200,000 births in the US a year. It's caused by a mutation in genes, including the NCF1 gene that encodes proteins that form an oxidase complex. And this enzyme kills bacteria and fungi to control infections. So if this gene is mutated, then the individual is more susceptible to bacterial and fungal infections. And just to speed up the talk, I won't go into it too much, but essentially, um, here's just highlights their preclinical data, just showing how it works in a mouse. And they got the prime editing efficiencies of the hemi, hemo, hematopoietic stem cells to be near 80%. So they can get it to get into cells and work really efficiently, at least in mice, and they have started the clinical trials for this. And now we're going to pivot to epigenetic editing. And this is I think, really exciting field. This one's kind of the newer of the ones we've been talking about compared to base editing and prime editing. So epigenetic editing targets the epigenome, epi meaning above, and that's this system of reversible marks regulating how DNA is read. So instead of changing the A, T, G, Cs, the letters of the DNA, the epigenome is kind of the methylation, and that can kind of make it harder or easier to read the DNA. So the expression levels of the DNA. And so you can imagine if researchers could make it harder for certain regions of the DNA to be read, then maybe they can avoid making any changes to the DNA at all. Maybe it can be a bit more reversible. Maybe you can turn like a dialing switch, you can just turn it down, turn it up. And so what they ended up doing is they took a DNA binding domain. In this case, they're Cas9. So your Cas9 with your guide RNA, and I guess you would actually use your dead Cas9 because you don't want to make a double strand break. And then you add the protein that will help modulate the epigenome. So in this case, you're using the DNA methylation effector domains or a histone modification domain. So as you've seen so far, all these kind of emerging tools are really just using the CRISPR-Cas9 protein or enzyme as a scaffold on which to attach other proteins. So you're in your base editing example, it's a deaminase. In prime editing, it's a reverse transcriptase. In this case, it's um, methyltransferase. So I think that's really cool, and researchers are getting really creative with all of these different tools. And so you can see on this cartoon on the right-hand side, people are able to silence genes by methylating the genes. They're able to kind of activate the genes, turn on the genes by demethylating them. And then they've actually been able to do both in a single dose of the drug. They can both ventilate some regions and demethylate other regions. So a pro or benefit of this approach is that it's reversible and non-permanent. It requires no double strand breaks, like I've said. Um, and you can fine tune the modulation of the DNA expression. The con is that it isn't permanent. Um, studies have shown that it can last a long time, but it's not permanent. So you might need to redose the patient. There are still delivery challenges. Like anytime you're attaching another enzyme to these Cas9 proteins, you're just making the complex larger, which can make it more difficult to deliver. And then it's still in early development, so we don't quite know all of the safety and efficacy associated with this approach. But the question, is it in the clinic? And the answer is it is about to be. So I've highlighted here Tune Therapeutics, and this hepatitis B is their focus for this particular therapy, and it'll be in the clinic actually in New Zealand. And this one uses lipid nanoparticles to deliver to the liver. And so what are they trying to do? For this one, well, hepatitis B, the virus, when it's in your liver, it replicates and then reinfects the liver. Uh, but hepatitis B cannot copy itself if essential parts of the virus are silenced. So the body has developed silencing mechanisms to kind of fight the hepatitis B virus. But unfortunately the virus has evolved around that. And so less than 5% of hepatitis B patients achieve full control of the virus. So what Tune is trying to do is kind of help patients deal with that mutation of the virus to go back and silence parts of the viral genome to prevent it from copying itself. Um, and then another company, Epic Bio, is seeking to begin human trials this year uh, for another heritable disease that severely weakens muscle tissues. And then also point out that Sangamo, which is another biotech company, is looking at neurological applications with epigenome editing. So really exciting field. And then before I move into the insertion of large genes, I'll finish up right here with RNA editing. So we've talked up into this point about how we're manipulating the genome and DNA. Well, what if we could make targeted changes to RNA? Well, people have been working in that domain for actually many years up until this point. If you know siRNA, small interfering RNA, that's a way people have been for a long time now, augmenting or changing transcription levels in human cells. But people are trying to use Cas9 proteins and this other ADAR protein to do it in a targeted fashion. So I won't dive into this too much, but essentially the benefits of using RNA editing is that uh, it doesn't run the risk of incorrectly editing the human genome because RNA is more transient. Let's say there is an off-target effect or a mistake, the RNA will eventually just get degraded. Uh, And then because CRISPR relies on bacterial enzymes such as Cas9 to cut DNA, they can provoke the immune system. Some RNA editing approaches actually use human protein, so you can avoid that immunogenicity risk. So with ADAR, it can be used to change a G to an A in mRNAs. So it uses a small guide RNA that guides the ADAR enzyme to make that change. And then the Cas protein, we've talked about Cas9, but there's other Cas proteins, so like Cas13 is a Cas protein that uses a guide RNA to target and cut a target RNA. So you just switch out Cas9 with another Cas protein and begin to make breaks in RNA or excise portions of RNA. And then, so has this entered the clinic? The answer is yes. So Wave Life Sciences was the first group to therapeutically edit human RNA back in October 2024. And I won't go into the nitty-gritty of the disease they were targeting, but it's a rare disease, AATD. It's an inherited genetic disorder caused by a single point mutation. And this mutation leads to the aggregation of proteins in the liver in hepatocyte cells, which can eventually lead to progressive lung damage. So about 200,000 people in the US and Europe will have a severe form of this disease. And what they were trying to do is they're using that ADAR protein. So like I said, this ADAR approach uses a small um, uh, kind of targeting RNA. In this case, it's that GalNAc AIMR and which binds to the ADAR, which then targets itself to the target RNA and makes that A to I switch. And actually, I's are read as G's. So that's kind of an interesting fact. When the nucleotide is changed to an I, the cell reads it as a G. So essentially, you're making an A to G swap. So now we're going to pivot to insertion of large genes. And this is a field I'm really excited about because let's say you want to insert an antibody into someone's cell. You're going to need to find an approach that can insert large genomic sequences. So one way to do that is using just transposases, these jumping genes. And so this graphic shows a researcher inserting a plasmid, and that's that circular thing that says a transposase, and then that gets transcribed and then translated into that SB means sleeping beauty, which is a type of transposase. So your sleeping beauty, then the researcher also introduced a plasmid with your gene of interest. Let's say in this case it's an antibody. So then the sleeping beauty transposase will cut the your gene of interest and then use that the transposase will use its insertion capabilities to insert into the genome. And yeah, transposases are super exciting. They've been around forever. They occur in many, many places across your genome. And so people are trying to use those and have been using those for a while to make large gene insertions. So what's new in this space? Well, one thing that's new is actually CRISPR-associated transposases. So researchers have actually found these naturally occurring in bacterial cells where CRISPR complexes, and so CRISPR-Cas9 is one version of CRISPR, it's a single enzyme, but there are other versions, some that are multi-subunit. So these CRISPR-associated complexes, some of them occur naturally with transposases, and this is really exciting because the CRISPR complex is what's used to target the genome, and then you have your transposase there um, inserting a gene of interest into the genome. Now, right where this field is right now, these were found in bacterial cells, and so now researchers are kind of engineering them to make them work better in human cells. And this is just an example of what researchers are doing. All those red dots on this, I forget if it's a crystal structure or cryo-EM structure, are points where the complex was engineered to improve its efficiency. Another approach are retrotransposases. So this came up previously in that prime editing where the Cas9 was connected to a reverse transcriptase. But this is an example of inserting an RNA into a cell and using that to turn into DNA to insert into the genome. So this is from Tessera Therapeutics, and this is your retrotransposon bound to a RNA template, and then it finds a complementary sequence with that DNA, which is pink, and then makes a cut, and then it binds itself, and then the reverse transcription occurs. And then that newly synthesized DNA is then inserted into the DNA. So that's also a really exciting area of research. Tessera is doing some cool work. And then recombinases. So this came something a really cool platform came out of the Arc Institute in California a year or two ago called Bridge RNAs. This one's a bit complicated, so I found this video that will hopefully help us um, understand it a bit more. So here's a bacterial cell that has these jumping genes, these naturally occurring bridge RNA guides. So this bridge RNA, which is in that gold, and part of it is bound to the DNA target, and then part of it is bound to the DNA donor. So what you want to insert into the genome. And it brings those two into close proximity. And then you do a strand exchange to form what's called a Holiday junction. And so then you fully recombined your donor DNA with the target using just this bridge RNA, which helps um, that switch. So you can see that that RNA has a very unique structure, quite complex. So researchers are still kind of optimizing that technology. It's super exciting that also that it was naturally discovered in nature and turned into a tool pretty quickly. So now we're going to pivot to AI-powered design and machine learning. So AI penetrating everything. So how is it showing up in the field of gene therapy? So I found three examples that I thought were kind of interesting. There's many, many, many more. This one is the discovery of new proteins. So one way you can discover a new protein is, say you have a large protein data bank or a DNA data bank. Let's say you have your Cas9 sequence that you've been using and you want to discover more Cas9s. Well, look at the DNA sequence of the Cas9 you use and see if there's a homologous version or similar sequence showing up in your DNA database. But sometimes that's not the only way to do it because let's say there are other sequence Cas9 proteins out there that have the same structure but a very different DNA sequence, then you'll miss those. So what researchers are doing is they were using AlphaFold, right, which was made by Google DeepMind, to predict protein function or structure. So they're using more of a structure-based search. So even if these Cas proteins diverge very, very far away in their DNA sequence, just using their predicted protein structure, you can actually discover them. So researchers have used that to discover smaller versions of Cas proteins, which is useful if you're trying to deliver the Cas protein to certain cells, right? Because these um, delivery vehicles, whether they're AAV or lipid nanoparticles, have a finite size to them. So it can be really beneficial to find smaller enzymes to fit inside. Here's an example of using AI and machine learning for AAV capsid optimization. So this one, they were just generated a whole landscape of single codon substitutions, insertions, and deletions, and they kind of measured the functional properties, what did those insertions and substitutions do to the AAV to make it better or worse? And then they used a machine-guided approach to um, engineer or continually improve the AAV design, right? So AI and ML is built on data, so you do need a data set on which the models can learn. And so they made some initial changes to the AAV and then used the AI ML to, you can see from this graphic, the red were improvements, the white's neutral, the blues made the AAV worse. And yeah, at the end of it, that it outperformed AAVs generated by random uh, mutagenesis, which is a common approach that many scientists use, randomly inserting um, uh, DNA into the AAV or removing or switching out. But this one was more intelligently designed using AI ML. And then the final example I'll give, which I just thought was interesting was MonaBio actually has this interesting chatbot, uh, AI based chatbot, where you can actually ask it real-time questions about designing an lipid nanoparticle or troubleshooting. They can predict kind of how your design might do if you input the formulation and the model, and it can help kind of design your experiment for you. So I think, that's a you're going to see more and more of these kind of scientific and research tools and aids, kind of building off of the large language models that others are developing, and making them very specific in this case for lipid nanoparticle design. So I thought that was a cool example. And let's see, the last thing I'll touch on is delivery, and I wanted to leave time for some questions. And this one, rather than you could give a whole talk on delivery, so I kind of wanted to end it with where the field generally is. This one was more of a focus on non-viral delivery. Um, AAV and lentivirals are still kind of the leading delivery approach in the clinic, but a lot of exciting work is being done in the non-viral space. So this just gives you a nice snapshot of targets in the human body and what's being used to reach those areas. So already in the clinic, you have ex vivo delivery to T cells and hematopoietic stem cells. You have ex vivo delivery to pancreatic cells and systemic. So systemic meaning you inject it in you inject it in the blood, it can find its way to the target cells of interest, so they can do that with the liver. But where a lot of interest is and progress needs to be made, there's only limited delivery methods right now to the lung and spleen, and then a lot of interest, but little progress yet in the brain, heart, and muscle. And then HSPCs are listed because people still are working hard to, like, if you can inject it in the blood and it can find its ways to HSPCs. That's still quite difficult. But people, as you know, are really working towards that and making exciting progress. So hopefully over the next couple years, we'll see these hurdles overcome. And I will end with this slide. A lot of the different graphics I used came from the Innovative Genomics Institute. Like I said, I worked there for a bit, and part of my role was develop educational content. So if you want to dive in a bit more into CRISPR's use in agriculture and research and clinical trials, CRISPR ethics, I would definitely recommend checking that out. We made something called uh, CRISPR Caspedia, and then we made all these freely available graphics. And then we also made some really cool virtual reality experiences where you can dive in a human cell and do the sickle cell, correct the sickle cell mutation. It's pretty cool. And then that's a screenshot of the cover of that white paper I mentioned from the World Economic Forum, where some members of the GGTI community and we kind of pulled together different case studies from around the world of teams working on gene therapy, bringing gene therapy to their countries. And then most recently, we pulled together several authors also from around the world to write a review. And so that's also freely available online. And there's my contact information. Please reach out if kind of anything you heard today piqued your interest. And I know we covered a lot of ground pretty quickly. So if you want to dive deeper, I'm happy to um, send you some more resources to help you learn a bit more. But I'll stop right there and be happy to take questions.
Thank you very much, Kevin, for that very educative talk, and also for the those video illustrations that try and make very complex science more understandable to people who are not experts in the gene therapy field. I noted that in the approaches you have not mentioned any lenti-vector approaches, but you mentioned the adeno associated vector and a lot of the new methods you've mentioned about the improvements that we have happened in the CRISPR method, and very exciting, and also the nanoparticles. I just would like to ask a question. Do you see the lenti-vector based editing going to disappear in the future, and what do you think are going to be in your opinion, the most what we are going to use in the front line, what's going to be out there most successfully as we go to the future?
Yeah, that's a great question. Well, there's a couple of visions of if you ask this question to anyone, they're going to give you a different answer. But I think one of the most exciting visions I see and what I've heard people kind of aspire to is this almost library of gene therapy library where you have your condition, or let's say a patient shows up with a certain um, uh, symptoms, and then their genome is sequenced, and they figure out, okay, what part of the genome do we need to target? Okay, what's the best approach to do that? Should it be a lentiviral? Should it be an AAV? Should it be a lipid nanoparticle? Can we do it with base editing? Do we need prime editing? So they are building this kind of library of CRISPR, if you will, or library of gene therapy. Some people in California are imagining this and teaming up with different academic groups with companies to see how this could happen. But lentiviral in particular has come a long way and will continue to be part of this library. I think LNP is interesting because the large gene insertion approaches, like I was mentioning, some of them are you can only are all RNA based, so it doesn't require DNA. So in that case, using an LNP is a great approach. But lentivirals are pretty well established, and I think where innovation in that area will come from is um, are how many, what batch size of lentiviral are you manufacturing? And I think there might be another discussion later on manufacturing, but that's an area of innovation that I'm really excited to follow over in the next few years. So I know there's work going on at different academic institutions and companies to figure out, okay, if you only need to make a small amount of it, how can you do that in a cost-efficient way versus a large amount? And so I think viral vector production in that space, those are the types of questions that I think innovation will really make a big impact.
Thanks, Kevin. There are a couple of questions in the chat. I don't know that you're able to see them.
No, we can read to him.
Right, so I have some challenges with my.
Okay, I can read, Doctor.
Yes.
The first question says, thank you for the presentation. Is there a risk of new disease with base editing? How do you decide on which amino acid to use?
So I guess with pretty much every approach I've mentioned, there's a risk of incorrectly making an unintended change to your genome. That's why epigenome editing was pretty exciting because you're not actually altering the DNA. And RNA editing, it's more transient. For base editing, there's bystander edits, I think is what they call them. And so that's altering a base pair that's near the base pair you were hoping to edit. So yes, you can change a letter that you were hoping not to change. But they're engineering the base editing enzymes to make them more efficient and specific.
All right, the other one is about prime editing. What are the chances of success that the new insertion is added into the chromosomal DNA rather than the original DNA with the mutation being reincorporated? What's controlling or regulation integration of the new DNA into the host DNA?
I'm not sure if I understand the question entirely, but I think as it's designed, the chances are really high that the DNA that you've made from your reverse transcription of the PEG RNA will be inserted where you want it to in the host genome. Yeah, I'm not quite sure how best to answer that question.
Okay, the other one says, so CRISPR seems to be very powerful tool for evolving technologies in gene therapy. How easy is it to develop CRISPR to Africa?
Yeah, I think, and that's what I touch on in is World Economic Forum white paper and the Science Translational Medicine review, where that question is multifaceted. I think it's as much a kind of a infrastructure and yeah, just bringing the right because uh and I guess for those who are at the GGTI meeting recently, kind of hearing about patients' experience going through one of these procedures, it's not trivial in terms of kind of the infrastructure a hospital needs for in terms of the chemotherapy and then staying in the hospital and then having the cells edited and then all of that. So I think it's and in my white paper I try to flesh that out of how can you design your infrastructure in such a way where it's not just solely built around a single health condition, but you can design it so it's useful for CAR T, sickle cell, and HIV, and so many different conditions. But I think it should be and it can be, and it'll just take collaboration. So that's why I think that GGTI is so exciting, because you need passionate people and advocates, because the technology is moving in that direction. It's just you need the support behind it to get the infrastructure set up.
In that vein, Kevin, are we likely to see a change in the CRISPR method from using electroporation, or it is bound to stay?
Oh, what? Can you repeat that question again?
Electroporation of the cells during the transduction with the CRISPR. Is it something that is likely to change, or it's going to be an integral part, to remain an integral part of that CRISPR method?
The
Electroporation.
Oh, electroporation. So that'll be useful for any, I think, the ex vivo approaches. So when you're bringing cells out of the body, then electroporate them and then put them back in. Um, I think that could likely stay, although electroporating cells also can have negative effects on the cell. So I think people are also trying to avoid that, but then a much more cost-effective approach is in vivo delivery. And so that's why a lot of effort is being put in that space because it will drive down costs. But I will also say, I think, kind of going back to the question right before that, ensuring that these technologies reach a broader audience and patient pool around the world, I think also requires researchers to design their experiments using a whole host and wide range of uh, genomes, and oftentimes they'll focus on databases or human cells that don't reflect the global population. And so when you actually introduce these tech tools into human cells of people around the world, you might see off targets that you hadn't predicted because you're just looking at different genomes. And so I think moving these questions of global access further upstream into innovation, like from the beginning, increases the chance of success, safety and efficacy downstream for everyone.
All right, thank you, Doctor. We have another one that says, it's quite long, but I'm going to be reading in bits. In developing gene editing strategies against viruses like HIV, do you think it is more effective to target the virus itself? For example, excising or inactivating the provirus, or to edit the host cells such that disabling receptors like CCR5? What are the key scientific and ethical trade-offs between the two approaches?
That's a great question. Yeah. So for HIV, there's different approaches people are taking. I think Excision, the company Excision, was going after excising the actual virus. Others, people are interested in inserting antibodies, mutating the CCR5 locus. I think deciding on which approach is best, I think you might likely need a combination um, to achieve uh, the outcome you really want, the long-term outcome. And so I think it's going to take a combination, and I think you kind of won't know until you know. So I think it's worth trying all of them, working your way up in mouse, non-human primates, and then that sh shows safety than humans. But that's a really great question, and people are pursuing all of those options.
All right. There is another one that says, in an event you take T cells from a patient, engineer them in brackets, CAR-T, and reinfuse them. Are they still the patient's own, or they are a new biological product?
Oh,
In an event you take T cells from a patient, engineer them, in brackets, the person put CAR-T and reinfuse them. Are they still the patient's own, or they are new biological products?
I would say they're still the patient's own, and part of the reason I would say that is because every time your cell replicates, it introduces mutations. And so the person you were maybe when you wake up in the morning versus when you went to bed, there are many, many, many more mutations in your body. So I guess you're the same. It's the same person.
Right. Dr. We just wanted to inform you that all these questions are going to be collated in one document. Then you will re-answer them, whether you've answered them or not. But still, let me read one more, then we give a chance to Adrian Wright to ask his own question. Is this hand up? No, we have, okay, we have a person asking, during the research, have you ever thought of creating epigenomes for modification? And if so, how effective can they be in eradicating such genetic cases?
So is the question just how effective is epigenome editing?
I think they're asking how effective it is.
Yeah, well, it's proven very, very effective in preclinical research. At least when I first saw the data, I was just surprised at how long lasting it is. And also epigenome modifications can even be passed down through generations, depending on which methylation marks you're looking at. I think it's a really exciting area of research that could lead to some fruitful outcomes. I think the main questions to consider are what does off-target edits look like and how problematic are they? Because we know what that is when dealing with nicking or double-strand breaks, but maybe not so much with epigenome modifications. And then, yeah, I guess dealing with the heterochromatin DNA that's kind of folded up or exposed. What does that look like in different cell types across the body? So I would imagine epigenome editing will have different efficacies based on the cell types you're interested in.
We had someone's hand up. Mutes, please kindly ask your question. Mutes, unmute yourself. We're waiting for your question, please. Your hand is up. Okay, let's have Mr. Luswata to ask. Okay, as we're waiting for them to unmute themselves, we have another question from Adrian. He says, great content question. In the limited success with delivery to target organs such as spleen and lung, what approaches was used to these circumstances to overcome the tissue or organ specificity challenge? Then it went ahead saying, what is the main hurdle in these cases? What are the other tissues, stroke organs listed as difficult, still a challenge?
Yeah, so I'll I guess give a response to maybe one response to each of those. One exciting area I've seen to help target the lung is what we touched on near the end with the AI ML. I think there's a company, Nanite, that is using polymer nanoparticles, but some people, I think Monobio and others are using more LNPs. They will have their delivery vehicle, put it into a mouse or a non-human primate. They'll see which ones make their way to the lung, and then that forms their data pool, and then they'll do it again, get more data, and then they'll start training their models, their AI models, to figure out like what about these particles that make their way to the lungs are actually driving them to the lungs. And so some companies are making some pretty interesting progress using that approach. But then your second question, I think the blood-brain barrier, I think a lot of people, you're ideally not going to cut a hole in someone's skull to inject this to deal with, say, Huntington's disease. Even though people have uh, shown that you can use CRISPR on Huntington's disease in mice, you ideally don't want to cut a hole in the skull. So wouldn't it be great if we could inject it in the bloodstream and have it pass the blood-brain barrier and go into the brain? But that's been proven very difficult.
What are the current delivery mechanisms for prime editing?
Um, I've seen well, the prime editor is so large that I've seen that you need to use dual delivery. And I want to say I've seen either having to deliver two AAVs or an AAV and an LNP. I don't know if I've seen the delivery of just one LNP, although I haven't been tracking like the whole landscape. But from what I've seen, people have had to use two delivery vehicles. Yeah.
There is a person's hand that was up. She has unmuted herself. Motez, please go ahead. Naomi? Naomi Mutesi and Mr. Tony Luswata, you had your hands up. Okay.
So do we have any other questions?
So this is all very straightforward and it's easy, I hope. But I'm happy to answer any questions. Please email me and I'll send resources.
So thank you very much, Kevin. I'd like to announce that our next webinar will be on the 22nd of September. So please do plan to attend. I'm sure it will be, another exciting learning period for us. There's an evaluation feedback, which is projected by the IT team. Please do respond to the poll. Otherwise, I'd like to thank all of you for participating. And Kevin, thank you very much for a great job done today.
Thank you for having me. Here, I can, Sissy, I can share the white paper right now.
Not from the center, but I think she can, Sissy, I think you can. Can you allow Sissy to give us sound and picture as well? Are you able to hear? Are you unmuted?
I think Dr. Sissy went off.
Yeah.
But we'll make sure we pass on this link to her because she's the one who did that white paper.
I just posted the link in the chat if you got it.
All right.
Great. Thank you all so much.
Thank you.